Okay, hey everyone, I'm gonna say something before we start so that Mia has her full time. First, I want to thank you all for coming for the on developmental bias and the interaction with evolution, with selection. We organized this, me and Howrun. I'm Diogo. Please come over and talk if you want to discuss this. We're all very excited for the talks today. And me and Howrun wrote a short perspective on this that's been pre-printed. And please look it up. If you have any comments, we'd love to discuss it with you guys. And welcome. And now I'm going to turn it over to Mia Akbar. Thank you so much.
Thank you to both of you for organizing this symposium and for allowing me to be a part of it. Today, my name is Mia Akbar, and today I am going to show you some of the fun things that I got to do as part of my master's thesis at Queen's University in Kingston, working with Dr. Rob Colati. But before I do that, I want to acknowledge that, I'll start by saying that I work with purple loosestrife, Phlythrum salicaria, is introduced to North America. So whenever I give a talk about purple loosestrife, I like to acknowledge that this research was conducted on the land of the Haudenosaunee and Anishinaabe. And as a settler scientist, I'm committed to reflecting on my ongoing land relations, and I recognize that settler colonialism and invasive species are inextricably related, and that humans have an obligation to consider the potential gifts, but also responsibilities that non-native species bring. In particular, I really am trying to expand my worldview to think beyond the native and non-native binary of introduced species that we often see expressed in dominant conservation science, and embrace alternative methodologies guiding ethics and frameworks put forward by the keepers of this land. We expect trade-offs between age and size at maturity, or life history theory predicts those trade-offs, where maturing early is often advantageous because organisms can increase the probability of reproduction before mortality, but it often means sacrificing on the larger size and increased reproductive output that comes with taking your time before maturation and accumulating more resources. Different environmental stressors, of course, may favor alternative values of this trade-off, and local adaptation models predict stabilizing selection of local optimum values within populations and shifting values of that optimum along environmental stress gradients. However, these models often focus on a single gradient of stress rather than the interaction of multiple stressors as predicted in natural systems. In flowering plants, latitudinal clines in growth and reproductive phenology are very common, such that early flowering at a small size is often associated with polar latitudes where there are shorter growing seasons, and taking a longer time to get big vegetatively and flowering later is associated with southern latitudes where the growing season's longer. And common garden and reciprocal transplant experiments often find this to be locally adaptive. However, the extent to which this phenological and size adaptation to environments that vary consistently with latitude limit or facilitate evolution to biotic stressors that do not always vary consistently with latitude is still kind of an open question that I've seen some really cool talks about this conference. So it's possible that adaptation to a fine-grained abiotic gradient may limit the ability of plants to respond to biotic stress, especially if there are strong constraints imposed on the developmental phenotype. So here, this is an example where a mid-size, mid-flowering phenotype would have the fitness optimum, but if mid-season herbivory or late-season seed predation select for later or earlier flowering plants, respectively, this may change the shape of the fitness function. However, this may also be observed if insect herbivory selects for smaller genotypes that are more associated with chemical resistance, because sometimes there's a trade-off predicted between chemical defense and growth, or if larger genotypes are more tolerant to herbivores simply because they have larger vegetative resources to defend themselves, or to tolerate, rather, insect herbivory. However, response to biotic stress may also limit the ability of plants to adapt to abiotic gradients. One possibility is that biotic selection constrains developmental adaptation to that abiotic gradient that I was talking about in the last slide. If random and patchy herbivory that does not vary predictably with latitude alters selection on flowering time by reducing the cost of the maladaptive phenotype. And what I mean by that is, in this case, the optimum doesn't change, but the fitness landscape flattens and decreases the efficiency of natural selection. So in this way, there are multiple ways that both abiotic and biotic stressors can interact to impose constraints on developmental phenotypes, facilitate rapid evolution and population differentiation, or a combination of both. And that's definitely the case for my friend purple loosestrife, who you all have seen if you commuted here, because it's in peak flowering right now. It produces these great big purple inflorescences, and it's all over roadside ditches right now. So if you've been out for a drive on the highway, you have seen this plant. And if you're from Europe, you've seen this plant because it's native to there. Anyways, it was introduced to North America in the 1800s. Or earlier. That's just the earliest that we have records of it. And purple loosestrife generally grows in an apically dominant way. So what I mean by that is, once a primary stem shifts to reproduction, once that stem flowers, we very rarely see any more vegetative growth from that plant. It's just shifting to flowering and developing buds and flowers, and then if those are fertilized, converting to fruits. And what we see is that plants from the northern end of the introduced range, where it's colder and the growing season's shorter, stay small and flower early, just like we would predict in those local adaptation models, in order for plants to complete a full life cycle before the first frost. And at southern latitudes, they take a longer time to flower and get nice and large vegetatively before making that switch. And previous common garden experiments and reciprocal transplant experiments have again demonstrated that this is locally adapted to season length, evidenced by those experiments. So purple loosestrife garnered a reputation as a highly invasive species in the late 90s, and in the 1990s, a biological control program was established, such that 2 specialist leaf beetles and 1 weevil were introduced from the native range. However, the limited follow-up studies that have been done indicate that the responses to this biological control program have generally been pretty variable. In this particular figure, we have years after release on the X and percent cover on the Y of purple loosestrife in a particular strand, and we see that there's a general decline, but overall there's a lot of error. And not as many consistent trends as we might expect, such that this is evidence or gives us an indication that there might be some rapid population differentiation for growth, development, and herbivore tolerance and resistance. So because of its developmental trajectory, rapid evolution and introduced environment and recent biological control program, purple loosestrife is this really cool system to ask questions about rapid adaptation to new environments and how that might be facilitated or constrained by abiotic stresses along environmental gradients, and how ecological complexity added by insect herbivores upon that subsequent introduction of the biological control program to the introduced range may facilitate or constrain this adaptation. So, if chemical or phenological resistance to herbivores trade off with growth and reproduction, we might see crossing reaction norms where genotypes that have higher fitness in the presence of herbivores are less competitive in their absence, with little flowering time and size variation between those treatments and maybe some between genotypes. Alternatively, if release from herbivory results in a demographic release and relaxed selection, we would expect to see higher fitness for all genotypes in the absence of herbivory, with more flowering time and size variation between genotypes than between treatments. To answer these questions, we used a common garden experiment where a previous master's student in the lab, Eugene, ambitiously sampled plants from 20 populations along 10 degrees of latitude in northeastern North America, ranging from Timmins, Ontario, down into the mid-latitude United States. And he selected 10 maternal seed families from each, and then planted them in 12 experimental blocks at the Queen's University Biological Station, where half of those plants were sprayed with insecticide and half of them were left to be subject to insect herbivory. And each year we measured growth and development of primary stems weekly between May and August. And at the end of the growing season in September, we harvested whole fruiting structures as a measure of fecundity. And now what I'm about to show you is results from those 20 population, or the source populations that we sampled, but I condensed the figures into means between regions because they're a little bit easier to look at that way. All that to say is that we found no evidence that herbivore resistance and tolerance trades off with size, which was confusing at first because, like some of the other plant herbivore talks I've seen over this conference, I had all of these big predictions about trade-offs between chemical and phenological resistance, and then there were no latitudinal clines. Great and cool. But in general, northern populations weren't more tolerant to herbivory, or they were technically more tolerant to herbivory by the definition of tolerance as a difference between fitness between the sprayed and unsprayed treatments, but this is more a function of the fact that those northern populations were constrained to flower smaller, so they just had less to lose in the absence of herbivory. And in coinciding with that, we see that in that sprayed treatment, those southern genotypes have increased fitness by like 50% compared to some of the northern genotypes. Ooh, wrong way. We also found that flowering time measured as growing degree days was significantly delayed in the presence of herbivory, but size at flowering was remarkably consistent between treatments, which I thought was really cool, with most of that variation clearly being explained by genetic differences among populations. So there's no ontogenetic change in flowering between treatments. Absolute flowering time is different, but not relative to those ontogenetic changes. This is consistent with a model that predicts there's a heterochronic style of variation for flowering time, such that shifts in flowering time arise when plants flower earlier developmental stages in terms of a population differentiation perspective, whereas flowering time differences between herbivore treatments simply just has to do with likely changing growth rate. This translated to an observed correlation between flowering time and size that's weakened in the presence of herbivory. So these are population means between treatments. And then to measure selection on correlated traits, we computed a principal component analysis. So because these traits are highly correlated, we use PCA to transform them into two uncorrelated axes for us to measure the strength and magnitude of phenotypic selection, where PC1 is the first axis of a PCA for flowering time and size, where individuals that have high values of PC1 flower late and large relative to individuals that have small values of PC1 that flower early and small. And then PC2 is the second axis that represents variation orthogonal to that expected flowering time and size correlation. And using a Landian-Arnold method to investigate phenotypic selection, we found that in the presence of herbivory, there was no significant selection acting on PC1 and reduced selection acting on PC2. And in the absence of herbivory, there was directional selection for PC1 such that genotypes maturing at a higher vegetative size and later flowering time were favored. In summary, there was no evidence that herbivore resistance and tolerance traded off with growth and reproduction. The consequences of insect herbivory are higher for southern populations that have more to lose, but that's not necessarily adaptive, or doesn't translate to adaptation of northern populations. And developmental constraints on size at flowering imposed by that adaptation to predictable latitudinal variables means that fecundity is significantly reduced in the presence of herbivores. And this provides evidence towards that second hypothesis that I was getting at, that herbivory reduces the efficiency of selection by abiotic environments by decreasing the demographic consequences for maladaptive phenotypes that flower at smaller sizes. And this, to loop back into the symposium about the interaction of developmental bias and selection on shorter timescales, I think that this is evidence that adaptive evolution and of developmental thresholds is constrained, or the response to selection is constrained by other environmental stressors like herbivores. That's all I have today, and I would like to acknowledge my supervisor, who's been an excellent mentor to me, and also the fact that I'm one master's student and I'm presenting 6 or 7 years of data. I did not collect that all myself. In fact, 21 undergraduate students and 4 graduate students did that in the field, and I got to help, but I also got to clean them all. But it would not have been possible without these people and all of my excellent supportive lab mates, so thank you to them. And I think I have time for one question.
Sweet. Well, that was like way cooler than what I'm going to tell you guys about, but yeah, that was great. Yeah. So what I want to start with is sort of a piece of art that I drew that I find that as kind of a good way to kind of think about my science and orient myself with what my questions are. And sort of the question that I came upon was, if you look at the picture on your right, it's kind of posing the question of how do you get a genome? How does a genome evolve to code these wildly different organisms in it, right? If you had no idea what metamorphosis was, you would see a larva, a pupa, and a butterfly, and you'd say those are totally different organisms, right? But that's all packed into the same genome. And what I kind of, question I want to pose, and we'll come back to it in a second, is how does that evolve? So this type of this phenomenon is very common across. I would actually make the argument that everything kind of undergoes some extent of metamorphosis, and we can talk about that if you don't think so. But ranging from insects, vertebrates, parasites, even. So this is a very common phenomenon, and we generally call these complex life cycles or a metamorphosis. So the idea that I'm kind of playing with here early in my PhD is that if you can understand kind of the basis of life cycle evolution, then that could kind of lead to a more general understanding of patterns of biological diversification. Because if you look, deeply rooted divergences are often associated with principal life cycle alteration. So that's an idea I'm playing with. I'm still thinking about what I think about that, but I think it's an idea that's cool enough to pursue. So getting back to that question of, well, how do you get all of those distinct phenotypes packaged into the same genome? You can kind of flip the script and say, well, why wouldn't you? Right? Why wouldn't the phenotypes presented by different stages diverge? And kind of the general idea is that there's pleiotrophic constraints, right? So if you have variation that's expressed during one particular stage, that maybe it's neutral, maybe it's beneficial, but that has deleterious consequences for another stage, well, that's going to be constrained, right? That variation is not going to be able to persist. So theory suggests that, well, life cycle complexity is kind of what you get when you break these pleiotrophic constraints. That's sort of what we're seeing when we see these crazy metamorphoses. And the idea is that this creates evolutionary independence between stages. So where I kind of found myself when I was approaching this literature is that we have this idea that, okay, well, you get complex life cycles when you break pleiotrophic constraints. But then the question was, well, how? How do you break pleiotrophic constraints, right? So that was sort of the question that I wanted to address and that I'm getting acquainted with kind of here early in my PhD. So here's sort of an idea, and I kind of got this from cell differentiation theory, and if anyone here is into that, I'm representing it as it was 10 or 20 years ago. So bear with me. But consider this model where we have a single gene, okay, and it has sort of this broad expression pattern across stages. And what you'd expect is some degree of evolutionary constraint that's determined by variation in this gene. But if you duplicate that gene, well, that's, in theory, alleviate some of the constraint on one or both of the copies, and you're able to get more divergence, both in function and expression pattern, across stages. So you can see after this duplication, the patterns of gene expression, you're starting to see more differences. So you get that function that's sort of expressed at one stage, you can get a little modification of that function that's expressed in another stage, right? And if we stack this up over evolutionary time, we start to see that more duplication, more divergence, you get more of this specialization in kind of the modifications of the functions that are expressed between stages. So this was kind of the idea that I put together, that I thought, okay, well, if this is sort of how it works with how you get differences between cells, then maybe that should scale up to be this is how you get differences in the phenotypes expressed at the organismal level, right? So there's a series of predictions that you would have with this, is that the more duplicates have diverged from each other, the more dissimilar their expression patterns will get. I'm going to use the term homologous group here instead of gene family, because I know that gene family has a very specific meaning to some people. So I'm going to go with homologous group. But basically, the more a homologous group, a set of duplicate genes, has diversified, the more different patterns of expression you're going to get, the more stuff going on with that kind of little modifications of that function. And then finally, that duplicate genes have more stage-specific patterns of gene expression. So you get more temporal specificity in when it's expressed across ontogeny. So what I did was pretty cut and dry. It's a very, very simple time series transcriptomic study. I just sequenced RNA across the monarch butterfly. That's the model system I happen to be working with right now. Across their life cycle, and here's just a kind of quick principal component plot that shows, hey, it worked. We see those differences, that we see that transcriptional decoupling between stages. So now what we want to go on and test those predictions with this data. So just to reorient everyone, so first thing we're going to look at is more diverged duplicates have more dissimilar patterns of expression. So we can look at this sort of, man, those are supposed to come up. Oh, never mind. So this is sort of, so what I'm going to do is I'm going to show you a few examples. You know, I have like 1,000 or so sets of homologous genes. So I'll show you kind of some examples we pulled out, and then we'll look at that distribution across all of the sets, right? So generally what we see is okay, so the top one here is a set of, relatively conserved or so Hox homologs. We see that, okay, yes, the x-axis here is the phylogenetic distance, so the phylogenetic divergence that homologous group has undergone, and then the y-axis is the expression pattern divergence. So we see generally, yes, with these two examples at least, that those two features are positively related. And then the bottom one is a set of Osiris homologs. Those are, used for cuticle development, which is something else I'm kind of interested in. I digress. But we can look at this distribution across, so look at these correlations across all of the 1,000 or 1,300 or so groups we have. And when we see that distribution is positively shifted. So if you look at where that cumulative density function crosses 0, everything to the left of it is a negative correlation, 0 is no correlation, everything to the right is a positive correlation. So generally what we see is that this holds up for the majority. It's like 28 or 78% or so of the homologous groups. Okay, so the second prediction that we're going to say, so this should be kind of an extension of the first one. If we're saying, okay, well, the more divergence you get within a homologous group, the more their expression patterns diverge. Well, then we can do comparisons across homologous groups and say, okay, well, the more diverse homologous groups should have more diversity in their expression patterns. They should have more different things going on with those kind of various alterations of the functions, right? So we'll see what this looks like, and again, we'll look at a few examples, then we'll look at what this looks like across all of the groups. And what we see is that, so here's an example of a geranyl synthetase-like group. Yeah, I don't know. But basically, this represents sort of a group that hasn't undergone very much divergence. And what we see is that the expression pattern, so on the right, and here we're looking at branch lengths, right? So we're looking at total phylogenetic divergence that this group has undergone. And we see that the expression pattern isn't very diverse. There's not a whole lot of going on. There's not a whole lot of that stage specificity, that temporal specificity that we're looking for, right? And we can contrast that with with another group that's undergone quite a bit more diversification, and we see that there's a lot more going on in those expression profiles for those groups, right? We do start to see more of that stage specificity. We see some of the genes in the groups are active in the larvae, some of them are active in the pupae, some are active in the adult, and we see a lot more variation in the temporal expression patterns. So we can look at what this correlation looks like across all of the homologous groups, right? And we see generally that yes, that the amount of phylogenetic, the amount of sequence divergence that a homologous group has undergone is generally a pretty good predictor of diversity in their expression patterns, right? So that's that's good. Okay. So the final thing is that duplicate genes have more stage-specific expression patterns. And what I want to show you here is just a simple distribution. We can compare the cumulative densities of singleton genes versus duplicate genes, and we see that distribution, so tau is just a measure of how, I think of it as how spiky that expression pattern is, because that's what we're looking at across the time series. We see that yes, duplicate genes tend to be more stage specific in their expression pattern, meaning they're more isolated to either a larvae or a pupal stage or an adult stage, stuff like that. So in summary, we found support basically for all those predictions. I'm gonna gloss over that. And now I wanted to kind of touch on like future directions. So one future direction I have is that is to question how general these findings are. So I'm looking at it, kind of trying to apply these same dynamics towards explaining the differentiation that a monarch parasite undergoes, and this is like a way diverged group, so that's kind of cool. And then I wanted to kind of pose some challenges. So one of the things that I've totally ignored here is regulatory element evolution, right? And that upsets me. It might upset some of you, right? Like we just totally ignore that whole aspect of gene regulation, right? So in the future, I want to try to like kind of build in and quantify the relative importance of like, say, a raw duplication versus regulatory element evolution and stuff like that. First paper's now out in Evolution Letters, so I encourage you to go read it. I left out a ton of nuance. And with that, I will take any questions. Yes.
Would you expect to see these same patterns in species with less dramatic
So my approach to kind of looking at this, that I thought that same question, and I was thinking of taking a comparative approach. So like if I'm saying gene duplication is kind of the source of variation for how you get transcriptional divergence between stages, then a comparative approach of things that maybe undergo less transcriptional divergence across stages, I think you would, what I would expect would be patterns of duplicate retention, right? I mean, theory suggests that kind of the rate of duplication should be kind of constant, but duplicate retention within groups that have what are generally considered more complex life cycles perhaps would be higher. So a lot of cool pop gen stuff going on with like structural variants that I'm excited about. But yeah.
Are you defining your homologous groups? Like, are you using a notation? Are you using
Oh yeah, so it's an alignment-based approach. So you have this issue with like when you're inferring homologous groups, you can kind of you have this question of like, well, how far back, right? How far back are you going to look, right? So part of that, so it's kind of like a self versus self blast of based off sequence similarity, and then we reconstruct the phylogenies, and then I did that across like a very strict threshold and a very lenient threshold to kind of capture what more inclusive and getting kind of more past more distantly related homologs within these groups would do. So yeah, it's blast versus blast and then phylogenetic reconstruction. So yeah, yes.
I wonder how do you calculate like the stage specific or temporal specific?
Like tau.
Like the expression specificity.
Yeah, I mean, there's like a formula for it in general. So it's an equation that's widely used when looking at like people study. Like I said, this was all derived from kind of cell differentiation theory. So it's used for like cell type specificity in general, and then a lot of work has kind of found, okay, well, cell specificity generally correlates with temporal specificity. So I don't have the equation off the top of my head, but it's a pretty widely used metric.
So I'll start by thanking the organizers for putting this wonderful symposium together and for inviting me to present my work. So this was work I done as a postdoc in the Marianne Felix group in Paris in collaboration with Christian Brandl and Mark Blackster. So now I'll be telling you about the role of developmental biases in phenotypic rates. Okay, so now start by this landmark study of Yusuf Ueda on the tempo and mode of evolution. So what they did was a meta-analysis on phenotypic divergence here on the y-axis, and on the x-axis is the time of interval of this comparison. What you can see is two different modes of evolution. So across shorter time scale, so less than 1 million years old. So it seems that phenotypic divergence is kind of bounded, so it goes around this interval. And in longer time scales, there is a burst of phenotypic divergence. Okay, so this creates this Van der Waals pattern, so that in groups that are very different from each other, so there is this explosion of phenotypic diversity, and the probability of this burst increases with time. So here it's about 25 million years. So in the game of this, it's always to think about what are the respective roles of natural selection, historic contingency. So in here, usually the story is that there are different adaptive peaks that are shifting throughout time, and this is what creates these divergent phenotypic divergences. Okay, so, but I'll be talking about developmental biases today to see to what extent this can also explain these patterns of variation. And I'll be telling you about, so I think of developmental biases using the conceptual framework of the genotype-phenotype map. So here we have our genotypic space with a corresponding phenotypic space that development will construct. So in here we start with one single genotype that has its stochastic variance. And then with de novo mutation, so there will be the expansion of this genotypic space with a correspondent exploration of the phenotypic space. And so in quantitative genetics, this is termed the mutational variance, so how much phenotypic variance can be generated with the de novo mutation. So in this example, trait 1 is very easy to change, so it has a high mutational variance, while trait 2 is very difficult to change, okay? And this is what I mean about developmental biases. So there are just some phenotypes are easier to reach in a phenotypic space than others. And if you want to think what would be the null hypothesis for this, it would be just an isotropic exploration of this phenotypic space. Okay, so all different combinations are equally possible. Okay, this is always in the distribution kind of way. So this will set which phenotypes are possible to generate with mutation, and then other evolutionary forces such as selection and drift will then work on this variation, and we'll have the standing genetic variance within a population or species, so VG or G matrix, as we've heard countless times during this meeting. Or if we do among species together with a phylogeny, then we can have rates of phenotypic evolution. So just to highlight that, I'll always be talking about phenotypes here, okay? So in this correspondence between mutational variance and standing genetic variance has been observed before, so this linear association in different organisms and developmental systems. So here in the nematode vulva, in the fly wing, the famous work of David Hull. And today what I want to go into is in a situation where there is contrasting phenotypic rates. Okay, so then the expectation then when you have contrasting phenotypic rates is that there will be distinct developmental biases, or in other words, there will be an evolution of this mutational variance. So the variational properties of a given system will change, so then this will bias what can change across different evolutionary timescales. So now, and we do this using C. elegans and other nematode species. So this is just the lineage of the worm. So the cool thing about this, so it's easy to generate isogenic lines and to cryopreserve them, so you can compare ancestral and evolved lines very easily. And I'll be focusing on this group of cells here, which will lead to the formation of the vulva. Okay, it is important for egg laying for males to cross with. So this developmental system stems from a group of 6 vulvar precursor cells named P3P to P8P that can be found in a broad range of nematode species. So you can compare really the variational properties of these cells at 1-to-1. And these are established by the expression of Hox genes and a gradient of wind coming from the tail. And throughout development, so I'm not going through all the details, so there are different division patterns and signaling molecules that will establish that these central cells from 5 to 7 will establish the vulva. And these other cells, they have the competence to take over these developmental fates, okay? But so here 4 and 8 will divide one time and fuse with the syncytium, while P3P here can adopt one of 2 fates, okay? Either divide like P4P or not divide and not being competent and fuse with the syncytium directly, okay? So, but what you need to take from this is that this cell has a variable cell fate, so it's a stochastic cell fate, while these other cells have invariable cell fates. And we do these inferences of cell fates using the DIC microscopy by counting the number of nuclei and their position. And here you can see that there are 2 nuclei for P3P, so you divide it one time. Okay. So for the rest of the talk, I'll be focusing on these two cells, okay, P3P and P4P. So previous work in the Felix lab had seen that among wild isolates of C. elegans, so there is a high variation for P3P division frequency. So this stochastic variation it changes among genotypes, while P4P is rather invariant. Okay. So the case gets cooler when in the given group. So here O'Shia's, we have the exact opposite pattern. Okay. So P3P is invariable, so it doesn't divide, while P4P has a broad range of division frequencies. So then we asked the question if this was due to the evolution of developmental biases that can change different patterns across scales. So for this, we generated hundreds of random mutant lines using chemical mutagenesis from wild ancestors of Caenorhabditis and O'Shia's to probe for this. And what we did was to measure phenotypic variation at 3 different evolutionary scales in the 2 genera. Okay, so first at a mutational scale, we started with 4 different species, 2 of each group, and within each one of these 2 different ancestors, so just to control for genotype-specific effects. So we phenotyped all these different random mutant lines from each one of these sets together with ancestor replicate lines, so we have a measure of its stochastic variance. So secondly, so we did for the within species standing genetic variance, where we phenotype different wild isolates of these 4 species, and these two parts were done in a block design and were scored in parallel, so we didn't know what we were scoring. So then we can have a good partition of the variance. And then the third component was the rate among species, where we phenotype different Caenorhabditis and different O'Shia species, and together with the Blackstone lab, we put phylogenes into them so we can actually estimate the rates of phenotypic variance. Okay? So this was with a total of more than 85,000 individuals scored by the IC microscopy. Okay? So it was a bit painful, but we got there. So now what I'm going to tell you about, so it's the estimation of this heritable phenotypic variance across scales using a Bayesian generalized linear model. Okay? And we'll start by the among species variance. Okay? So just to put them into a phylogenetic context, so these 2 groups diverged about 80 million years ago. From these estimates are a bit on the lower scale. And what I'm going to show you now, so it's just a simple trait mapping along the phylogeny. So 0 red, so no division, and when it's blue, it's dividing. And if I start by P3P, you can see that on the Caenorhabditis part, so there is a lot of color change. I'll show you all the estimates of this. While in the O'Shia's part, it's essentially red, so no division. And when we look at the other cell, P4P, we observe the opposite pattern. Okay, so P4P is not changing its color, while here it's changing a bit. And when we do the proper phylogenetic mixed models, so here just an estimation of the increase in variance per million years. So here on the x-axis for P3P, on the y-axis for P4P. And you can see that among Caenorhabditis species, P3P is the one that is evolving the fastest, while in O'Shia's it's for P4P, okay? And if we put this in a log ratio, we can see clearly that among Caenorhabditis species, P3P evolves 3 times faster than P4P, and we observe the opposite in the O'Shia's. So now if we go to within species, we observe a very similar pattern, okay? Which is within O'Shia species, P4P is the one that is changing the fastest, while for Caenorhabditis is P3P, okay? So we have this, that within a given group, we have the matching within and among species, and it's contrasting between groups. So now the question is if this is due to the evolution of the mutational variance or developmental bias, whatever you want to call it. Okay? So if we start by Caenorhabditis, we do observe, as previously shown with mutational accumulation lines, that for the 2 Caenorhabditis species, it's P3P that is changing the fastest, about 1 order of magnitude faster than P4P. And for O'Shia's, we observed the exact opposite pattern, which is P4P that is changing the fastest, about 3 orders of magnitude faster than P3P. Okay, so we come to a scenario where across evolutionary timescales within a given group, there is an alignment between mutational variance within and among species phenotypic variance, and this is contrasting between groups. Okay, so thus we can predict the evolution of phenotypic rates across millions of years of evolution based on the evolution of these mutational properties of the system, and we have some evidence that I'm not going to show today because of time, that this is because of changes in this wind gradient from the two groups that makes them, this P3P cell or P4P cell, more or less sensitive to incoming variation, being genetic, stochastic, or environmental. So I want to thank everyone involved in the study, all our collaborators, Pierre, Henrique, and François for discussion, all the world sampling efforts from many different labs, our funding. And recently, so I moved back to Portugal in Lisbon, where I'll be working on the roles of developmental biases for behavioral evolution. And I'm very fortunate to be hosted by Sarah Magalhães and the MITE squad and Alice Sena. Thank you. Yeah, I spoke a bit too fast, sorry about that.
A great talk, just a very quick maybe clarification. It seemed like you were modeling the variability in the P4P, P3P as a continuous trait as opposed to a discrete.
Yeah, so yeah.
Was there a reason? I may have missed that.
Yeah, sorry, I didn't explain. So I used the I used the threshold model on them, so just the classical Wright model. So I have a liability scale. Okay, yeah. No, okay, good. So then I can use the exact same principles of quantitative genetics, so I just use the two different scales. Yeah. So I didn't want to go into the details. So I can tell you about the wind stuff then. Okay, so the dose response curve. So there are dose response curves to the amount of wind. So in Cinorhabditis, if you put different copies of wind, you can change P3P division frequency very nicely. And when you do the same thing in Oshayus, it's only P4P that is changing. Okay, so then the hypothesis was then that this gradient is changing, and by doing quantitative in situ, where you can actually count the number of RNAs, you see that correlation. Okay, so what we want to do next is to manipulate the wind gradient and see if the mutational variance actually changes. Okay, and so the whole idea is just I probably didn't explain that. So they are displayed. So they are in the anterior posterior axis. These cells. Okay, it's just being in a more variable part of the gradient or less variable of the gradient will make them more sensitive to incoming variation. So that's.
Does this mean that you can't change both of them at the same time? Can they both be high variance or no?
So what we have observed, no. So we not with this data set. So we did, or Christian did some nice experiments which instead of putting mutations, so he just used the same starting isolates and he put them into 40 or 50 different environments, okay, just to see how well they would match. And in the case of C. brixi, we do see in some environments that both cells start changing.
All right.
Cool. Yeah, thanks.
Okay, so for the final talk of the session, I want to welcome Patrick here. So please take it away.
Thank you so much for having me. This has been a very, this looks a little different than I thought. Anyway, it's going to be just fine. This has been a fantastic symposium. Thank you so much for having me. Yeah, my name is...
Check if the slides are working.
Okay, yeah. I just moved to San Diego where I started my lab, but I would like to talk about a collaboration together with David Berger from Uppsala University, which is a project that we have been working on for the past few years. But all of this really is motivated by this. Like, I'm fascinated by the amazing diversity and disparity that we see across the tree of life. And sometimes I catch myself thinking that evolution can really create any kind of imaginable organism there is. But that's of course not true, because both macro and microevolution often follow distinguished patterns. And we can see this here in these morphospaces, where even though, in this example, reptiles have been around for a very long time and they occupy a large component of this morphospace, they don't move into all of those spaces in the morphospace. And that has been recognized for a very long time. And it has been argued that maybe those empty spaces are just associated with low fitness. And that means that selection removes any species or genotype that dares to move into those spaces. However, similar patterns can also arise if the developmental architecture underlying those traits is just incapable or less likely to produce those trait combinations, and that's what we refer to as developmental constraint or developmental bias. Now, when I talk about developmental bias, that's what I'm thinking about. So usually we think about phenotypes as the result of a developmental process that is instructed by a genotype in a given environment. And if we introduce perturbations to this developmental system, for instance, due to introduction of novel mutations or environmental fluctuations, one might expect that this leads to random phenotypic effects. But as we have heard today, that's of course not true, and very few systems that's actually the case. So usually developmental systems are biased towards producing some phenotypes much more often than others, and that's what we call developmental bias. And this has been shown in many systems, most famously maybe a segment number variation in centipedes, but also birdwing evolution and many other systems. So we now have this accumulating data that suggests that some traits are easier to generate than others. And maybe those traits that are easier to generate developmentally are also those that evolve faster. And if that's the case, we might be able to use developmental bias to predict evolution on different timescales. In addition to that, which would be neat, I think this also asks fundamental questions about the role of development in evolution, which is why I'm really interested in those questions. So do we have any evidence for developmental bias predicting evolution on long time scales? Yes, I think we do. And this is just one example that we just heard about too, coming out of David Hull's lab, where they looked at macroevolution in Drosophila wing shape that accumulated over 40 million years of evolution. So what you see there is a sketch of a Drosophila wing, and you can see how different wing nodes accumulate different variations over the phylogeny. And what Hull et al. Found was that those genotypes that are easier to generate through developmental perturbations, in their case mutations, are also those that evolve the fastest. So in other words, those dimensions, those phenotypic dimensions that are most strongly affected by mutation also evolve faster. And that suggests that there's a strong developmental bias in flywings, and that this can predict evolution on several decades, several tens of millions of years. So now I'm wondering, do we find this in other systems as well? On what timescales do we find these relationships? And especially, what are the mechanisms behind this predictive power of developmental bias when it comes to evolution? So to look at that, David Berger and I, we teamed up and studied the black scavenger fly, Sepsis punctum. Now this is a small fly, they kind of look ant-like, but you can tell them from ants because they have only 2 wings instead of 4, and they have this little black dot close to the tip of the wing. Now to the untrained eye, this fly might look like a Drosophila, but I just want to point out that those two families have diverged about 60 million years ago, which is similar to the divergence time between an elephant, a hyrax, and Sirenian. So these are really 2 different beasts and 2 different evolutionary contexts we're looking at this at. So how do we measure developmental bias? We did not have the time, money, or nerve to do mutation accumulation lines. So what we did instead was we looked at fluctuating asymmetry. And I want to tell you why we think that this is actually a good estimate. So in bilateral organisms, the left and the right side of the same individual not only share the exact same genotype, but also the same environment. Yet we still see little deviations between the left and the right side, which are not due to measurement error alone. So we can quantify those differences, which arise due to random developmental perturbations that accumulate throughout development. And we can then test whether there are some phenotypic dimensions that are more strongly affected by those random perturbations. We did this in the fly wing, in the sepsid wing, and what you see here is a sketch of the outline of a wing. In black, you see the average location of particular landmarks, and in blue, I'm showing you the distribution of fluctuating asymmetric variation. What you can see is that all of those landmarks vary somewhat, which is expected, but there are some landmarks that vary more than others, especially the ones in the middle. I don't have a pointer. Those that are related to the positioning of the crossveins. So the 2 crossveins react most strongly along the proximal distal axis, on the x-axis from your view, when you introduce these random developmental perturbations, suggesting that they are the phenotypic dimension that varies the most, so they are developmentally the most variable, indicating a developmental bias. So having established that, we can now ask, does this predict macroevolution in this clade? And we did that by quantifying wing shape variation across 36 different species of sepsid flies, depending on whether you include subspecies or not. And we found that those dimensions that are developmentally most variable in this one species are also those that evolve the fastest across those species, suggesting, similar to what Hul et al. Found, that developmental bias can predict evolution on a macroevolutionary scale. And in fact, those patterns were so similar to Drosophila that we had kind of this crazy idea of what if we take developmental bias in sepsids and we try to predict macroevolution in Drosophilids? And lo and behold, it works. We can predict evolution in Drosophilids based on developmental bias in sepsid flies. And again, those are two, I think this is a stunning finding because those two groups have diverged about 60 million years ago. So this made me think, well, maybe this is just an intrinsic property of flywing development. So I wanted to look at this in many more species, but keeping species in the lab is really hard. So I leveraged 150 years worth of data in the taxonomic literature. So I went into the systematic and taxonomic literature and sourced a lot of high quality data from illustrations and both pictures. With that, we got data for about 900 different species, belonging to 43 different families, spanning about 185 million years of evolution. And with that data set, we can now ask, do those phenotypic dimensions that have most developmental variants also evolve the fastest on these timescales? And what do we find? Yes, they do. It's a little messier, the pattern, which is expected, but we can predict the major directions of evolution just based on looking at one single fly species, essentially, which is really confusing. Why the hell would we see an alignment between developmental bias and macroevolution on these kind of timescales? It's absolutely crazy. So there's one hypothesis that might explain this, which I will term the constraint hypothesis, which essentially suggests that developmental bias simply constrains evolution on a very long time scale. And this hypothesis has recently gained a lot of support by this really amazing science paper by Holstad et al. However, unfortunately, I don't think this is really likely to hold in flywings. And that's because wing shape evolution is way too slow. It's 10,000 times slower than expected by simply mutational input, unless all of this mutational input actually has deleterious pleiotropy and therefore is effectively unusable for evolution. Now, measuring deleterious pleiotropy is really hard, but we tried to do this by leveraging a common garden study that David did, which included 71 isofemale lines, and David measured painstakingly 5 different fitness components that are, by the way, not related to flying. They're not related to flight. And we get these breeding values. And what we saw was that small flies generally lay fewer eggs and they die earlier. Doesn't stop there, because their offspring are also small, they develop slowly, and they also have low survival. That indicates that overall we have this gradient in genetic quality or fitness along those lines, where some lines just perform better than others. And now we can ask, does this gradient of genetic quality correlate with wing shape, as would be expected if there would be the possibility for deleterious pleiotropy? And we really tried a lot of different analyses, but we really don't find anything. There's no relationship between wing shape, genetic differentiation in wing shape, and these fitness components that are not per se related to flight. So we don't really find a lot of evidence for the constraint hypothesis, and at the same time, we also have many instances of very rapid wing shape evolution, even on ecological timescales in various dipterans. So there really does not seem to be a hard constraint on wing shape evolution. So what would be an alternative hypothesis? Well, one of them is the congruence hypothesis, which suggests that maybe developmental bias and evolution align because they're both driven by similar patterns of selection. Now, flywings have a very similar function in all species. They're used for flight. And maybe that's why they all face pervasive functional constraints. Now, how are we able to look at functional constraints in thousands and thousands of species? I think this is going to be really hard, but there's some hints at least that we can look at, and that is when we look at the allometric scaling between wing shape and size. So if a fly gets larger, flight dynamics get different, and they have to somehow adjust their wing shape in order to still function. And we see a very large range of body size in flies, and that is correlated with variation in wing shape. So this is a pattern that we see across different species, but we also see very similar patterns within species. And interestingly, the actual shape changes that we see across species mirror those that we see within species, suggesting that there's a shared sort of potentially functional constraint on all of those. So now we can ask whether the developmental bias that we see in the macroevolution are aligned with these axes that are potentially under selection. And what we find is that indeed, both of those patterns are more strongly aligned, much more strongly aligned with allometry and potential functional constraints than expected by chance. And I just wanted to highlight that in both of those matrices, we actually accounted for allometry. So this is not just, you know, regressing one thing against itself. So with this, we conclude that developmental bias and evolution are both aligned with allometry, indicating that maybe functional constraints might be the driver behind this alignment. So to conclude, developmental bias predicts macroevolution over a very long time scale, and that has been very puzzling and has given me sleepless nights. However, this alignment is inconsistent with constraint hypotheses, at least in this particular system. Yet, there's some hints that maybe persistent correlational selection related to function may shape developmental systems such that they are more likely to produce adaptive variants. And if that is the case, we might have to reconsider the causal relationships between mutation, selection, and adaptation. However, I want to point out that even though we currently have very limited evidence for the constraint hypothesis when it comes to flywing evolution, we also don't have particularly strong evidence for the congruence hypothesis. So I'm really excited for the next few years to see what we'll find out when it comes to wing shape evolution. With this, I would like to thank David Berger, who has been a fantastic collaborator on this, and I also want to highlight Armin Mocek and Yong Hu, who have been collaborating on similar projects on dung beetles that I didn't have time to talk about, but we also had a lot of really insightful conversations about this. And yeah, thank you so much for your attention.
Hi, it's a super great talk. This may be I don't know, it's a question. So what if the functional constraint actually changes? Like, for example, I studied the diptern larval motor system, and the habitat changes, right? So there's always a functional constraint, but the functional constraint is changing. And so do you have any ideas for how that might affect, like, what if you were looking at larval motor traits instead?
So that's one thing that I really want to look at, because those flies, they have crazy diversity in different ecologies. And I would expect that those functional constraints do change if they really are functional constraints to begin with. And then it would be really interesting to actually measure developmental bias in different groups and then look at the evolutionary variance that we see within those groups to see if they align. Yeah, I think it's amazing. We actually, I mean, flywings are some of the most heavily studied. I mean, they're the best understood traits, really, of any metazoan, but we still don't know much about their selective pressure that they experience, especially in nature. So it's really, yeah, I don't know. But it's a great question. Yes.
As you said, I guess your evidence against the constraint hypothesis was not very strong. And there is some evidence that wing shape differences between the sexes do lead to evolutionary constraints. So I guess what kind of data would you want to collect to test your constraint?
So I think I guess it also depends on what kind of constraint are we talking about, right? Is it a genetic constraint, as in, do we not have enough genetic variation for it to actually evolve? Or can we not get that genetic, is it impossible for development to produce those things to begin with? I mean, our idea to look at deleterious pleiotropy, I think that was a good attempt. The problem is that we are looking at isofemale lines, which to some extent are inbred. So some of the lack of evidence that we see might also be related to just inbreeding depression, which might not be very relevant to the natural situation. So yeah.
So you'd want to look at it in outbred population?
I would like to, yes, to see how that goes. And then also the question is, how does developmental bias itself evolve across the phylogeny? We only compared that between sepsids and Drosophilids, but we have thousands of other species that we could look that up. So, yeah.
All right. Oh, one more question.
Do you have a model of what an optimal wing shape would look like for these flies and how similar the wings are to that optimal model?
I don't know. So for sepsets, we do have sexual selection data. We have, yeah, that's about it. We have sexual selection data. So for sepsets, in these particular contexts in the field and in the lab, it was actually fairly consistent between estimates in the lab and the field. Yeah, but I wouldn't be able to predict that for all of those different species because they have such different ecologies. So, yeah.
I'm just thinking, like, I imagine, like, aeronautical engineers might have ideas of, like, how wing shape would work well.
Oh, absolutely. I think in terms of, especially like the aspect ratio and those kinds of mechanical aspects have been studied really well. My problem is that there's so much pleiotropy between wing shape and the rest of the body, at least that has been shown in other studies, that I'm not sure, if you just look at flight, yes, absolutely. But then if you have all of the other pleiotropic effects, I'm not really sure how selection would act on that. But yeah, thank you.
Okay, thank you all for coming.